Good evening. My name is Bruno. I'm a gastroenterologist at the Erasmus Medical Center, Rotterdam in the Netherlands. And I welcome you at this PENTAX Medical Symposium, Scientific Perspectives on Hygiene Innovations in Endoscopy. As endoscopy, safety of our patient is at the core of our daily work and professional attitude. And infection prevention, infections caused by contamination of endoscopes, is becoming a more and more important part of that. In this symposium, we want to share some experience with you. And personally, I had some experience with regard to an outbreak in my hospital concerning 30 patients. I know what an incredible impact it can have both on patients and personnel. Over the years, some solutions have come about with regard to trying to prevent these contaminations, scope innovations, which we would like to discuss during this symposium. And for that, I'm accompanied by a few interesting speakers. For one, Ulrike Beilenhoff, the former president of ESGE and today's ESGE's scientific secretary, and Dr. Pineau, the director of Eurofins Biotech Germany. And for that, I would like now to give the word to our first speaker, Ulrike Beilenhoff, who would speak about the role of single-use consumables to improve patient safety in endoscopy. Ulrike.
Yeah, thank you very much, Marco, for the kind introduction. Single-use devices really improve patient safety in endoscopy. And my task is now to highlight the effect on our daily work as nurses as well. So that are my conflicts of interest. And when we compare reusable and single-use consumables, we have to take into account that we can use reusable devices for a prolonged time, but then we have really the problem with the quality and with damages and with less functionality. And people often forget that when they use, or people often think when they use reusable devices that they save waste. But reprocessing also causes waste, and we need material like process chemicals, machines, equipment. The reprocessing also costs money, and it is time consuming. And therefore, we have also the risk of infection, and not only for the patient, but also for the staff and potential health problems. We have the risk of recontamination, and we have, of course, insufficient function and the potential of damages for the endoscope. So if we use single-use devices, they are ready to use, they are sterile, and they always provide us the same high quality. But on the other side, we have to have the money for financing that, and we need extra budget for that. And this has to be discussed as well, especially the refund of all these accessories. So we need additional logistics and additional storage for these single-use devices. But the advantage is really that we do not need any reprocessing, that it is time-saving and it saves resources. And the most important benefit is really that it reduces infectious risk and enhance patient and staff safety. There are also controversial discussions about the waste disposal and the ecological footprint, and we can discuss that later. So when we now look at the different aspects, I would like to discuss with you which problems can we solve when we use single-use accessories. The first thing is logistics. We have a lot of challenges. Our endoscopy rooms, the reprocessing rooms in endoscopy units are often quite small. There is the potential risk of recontamination. And there is also discussion where should these accessories be reprocessed, either in endoscopy units or in CSSD departments. If we do it in CSSD departments, we have the problem with the transportation, again, the logistics, and also the question in which washer disinfectors can these devices be reprocessed. And then we have to answer the question, do we need sterile accessories, for instance, for the brushes? In endoscopy units and also in CSSD departments, there is really a lack of qualified staff. There is a high turnover, and there is often also the problem of lack of awareness. So if we now have a look at the valves, there are inconsistent recommendations in guidelines. they allowed the use of reusable as well as single-use valves, but single-use valves are not available for all endoscopes at the moment, for instance, for EOS scopes. And if you have a look here at the pictures, you see that the valves have a very complex design with narrow lumens, and you need multiple steps, up to 30 steps, to reprocess one single valve. And there is also reports from field studies and from surveys that staff really skip these reprocessing of valves, these cleaning steps, and we found gut germs, water germs, and also hand germs in reprocessed valves. And there were also outbreaks reported with valves, especially from the bronchoscopy area. And you see reprocessed valves also show a lot of damages. We also have the problem with documentation and with traceability. If we have a look at the brushes, guidelines nowadays recommend single-use brushes only, but there are still reusable brushes in use. And you see here these damaged reusable brushes. You also find residuals on the brushes. And even single-use brushes are used multiple times, which then cause, again, the risk of cross-contamination damages. If we reprocess endoscopes, we need different brushes for the distal tip, for the channels, for the valve ports, and also the time is not really defined. There are multiple brushing circles recommended, minimum twice brushing cycles. And again, if you use reusable brushes, you also have the problem with the documentation and the traceability. We all are aware of the outbreaks which have been reported since the early 2000s, not only from the United States, but also from Europe. And the endoscopy, the duodenoscope design changed within the years. First, we had just duodenoscopes with fixed distal tips. Then modules came up with a removable distal tip. And since 2019, the FDA recommended transition to a new design, and that really pushed the manufacturers to develop single-use solutions like this removable cap, but also solutions where the whole elevator system is included in the distal cap, like with the PENTAX solution. When you evaluate the outbreaks of the duodenoscopes, which were reported especially with multi-drug resistant germs, then you see that this is not only the small defects at the distal tip, but it is a variety of sources which caused the infection. It was insufficient cleaning, drying, and storage problems reported, non-compliance with guidelines, and also insufficient cleaning equipment like brushes were used. There were also field studies and surveys which reported that up to 75% of the staff said yes, they cancel reprocessing steps, especially the manual ones, when they work under time pressure. And then brushing and drying mainly were skipped. And there is also a problem with training and awareness. So guidelines clearly recommend that routine inspections should be in place after each reprocessing step, and also microbiological surveillance on a regular basis. So in conclusion, there are still controversial discussions about waste disposal, the ecological footprint, and also the financial impact and the refund. And we can discuss it perhaps later, but there is clear an advantage for single-use consumables because they enhance patient and staff safety, they solve logistical problems, they save time and resources, and they really ensure traceability. So thank you very much.
Thank you, Ulrike, for this very interesting presentation. What kind of strikes me, and I would kind of go back to some of the basics. You explained something with regard to efforts to clean a scope. Can you elaborate on that with regard to time and the many steps it takes to clean a scope?
Yeah, endoscope reprocessing is a very complex procedure, and it's really time consuming because you have the manual steps before you put the endoscope into the washer disinfector, and then you have really a certain amount of minutes for the automated reprocessing. But for the manual reprocessing, you really need time as a nurse, and that is difficult if you work under time pressure. I would say, depending on the time of the type of endoscope, you need between, let's say, 5 to 20, up to 20 minutes to reprocess, to clean the endoscope.
Yeah, and it takes more than 100 steps to complete.
Definitely.
So the least paid personnel in the endoscopy department has a very big responsibility with regard to cleaning scopes. Okay, Ulrike, you mentioned nicely in your presentation some of the solutions that we currently have in an attempt to improve patient safety, and the valves, the single-use valves, the brushes, and the tip, the removable tip. Do you foresee any new solutions that could aid to further improvement in this regard?
Yeah, from the workflow, it would be very helpful to have kits which combine valves, rinsing adapters, and also the distal caps, so that we have everything in one package, and also kits for the reprocessing, so that we have different brushes available for one endoscope. That would be very helpful.
And I would be kind of curious, because you mentioned this, and we have this available, these kind of solutions. If you look down at the market in Germany, for example, what is the penetration? How many hospitals now have converted to the use of these kind of disposable materials in endoscopy?
I think the single-use brushes are very well established. They are used all over Germany. The valves are sometimes a problem. There is clearly a trend towards single-use valves, but there is really a problem that they are not available for all types of endoscopes, especially for,
Right.
let's say, US scopes. No?
So there's still room for improvement there.
Yeah, definitely.
Okay, thanks for these answers, Ulrike.
I have also a question to you. You have a great experience as an endoscopist. Do you have also experience with these single-use consumables?
We have a lot of experience with these single-use consumables. We use it in our practice daily, and I think they add to our patient safety profile. And also now we have some experience with single-use endoscopes, which is a whole new development in this regard. For sure, it abolishes completely the risk of exogenous infections, but the transition towards this type of endoscopy is going to be relatively difficult from an economical point of view, of course, because these scopes are quite expensive. And also because there is a large capital equipment investment into the reusable scopes. And also because these new developments with regard to hygiene prevention makes the use of these reusable scopes safer. So with regard to these disposable scopes, I foresee a future, but it will take quite some time, and there is still a future ahead for the reusable devices. And for that reason also, we should focus our attention on making that type of endoscopy safer with all the means that you have so nicely and eloquently alluded to.
But there is still the problem of the availability that we have the single-use endoscopes, and also the problem with the refund, no?
Absolutely. And also from a production point of view, because even if you imagine that that ESGE will be done by disposable scopes, and so many scopes have to be produced, this will take years and years to become in motion. But in my personal view, this is something that we cannot revert anymore. There will be a gradual transmission with regard to the use of these scopes, but it will take years and years.
Yeah, but we already saw it in bronchoscopy, so but perhaps we will take the same route. I totally agree with you.
We should not forget, indeed, in urology and in bronchoscopy, already hundreds and thousands of these scopes are being sold at this point in time. So it's only time before this will also be the case in gastroenterology. Okay, thank you, Ulrike, and for making time. I would like now to introduce our second speaker, Dr. Pineau, who's a director of Eurofins Biotech Germany, and he will talk on the important subject of endoscope drying and the prevention of biofilm formation, and in this case, with the use of plasma typhoon and plasma bag system. Dr. Pineau, the floor is yours.
Many thanks, Marco, for this very kind introduction. So, as you mentioned, the main topic of my presentation is to give you a brief overview of the importance of drying to prevent biofilm formation in endoscope channel, and share with you our feedback regarding the test done on the plasma typhoon and plasma bag system. First of all, I would like to start my presentation with some data regarding the quality of endoscope. As you know, in fact, there is only few indication or data published on the real microbiological quality of endoscope. And in fact, we really don't know if we are doing a good job when we reprocess that. Some information we have in the lab is the follow-up of the quality of endoscope done in our lab by sampling on the endoscope on a routine basis in more than 180 public or private hospitals in France. And the results we have give us a good idea about the situation, at least in France, about the quality of the endoscope. What we discover is that in 2004, around 20% of the endoscope were at what we call the action level, meaning that they contain at least one pathogen bacteria or more than 100 CFU of non-pathogen bacteria, and that they could not be used, they should be quarantined. Even if we observe year after year that the result seems to be better and better, we still have in 2019 around 11% of endoscope considered to be at the action level, that means which cannot be used. Of course, we can discuss a lot about what could be the target value regarding endoscope contamination level, but I will not spend any more time on that point. The other interesting point dealing with this study is that we found about 37% of the microorganism or 37% of the microorganisms isolated from endoscope channels are waterborne bacteria. It's something really, really interesting. And because these waterborne bacteria are able to form biofilm, it's a real problem for endoscope. The paper published by Pashkos in 2004 confirmed that the presence of biofilm in endoscope channel is a real problem. Pashkos also mentioned in his paper that routine cleaning procedures do not remove biofilm reliably from endoscope channel, and that this may explain the unexpected failure of reprocessing procedure despite good adherence to guideline. But also Pashkos mentioned that the key point to eliminate biofilm, or more to avoid biofilm formation in endoscope channel, is the drying of the channel of the endoscope. The importance of drying is also highlighted in many papers or recommendations. Thus, in the well-known study published by Kovalida, the author mentioned that accurate endoscope drying is critical, and that the final drying steps greatly reduce the possibility of recontamination of the endoscope by waterborne bacteria like Pseudomonas species. In the other paper present here, published by Musarella 2 years before, you can also see that endoscope drying as well as alcohol flushing in some country, followed by forced air or compressed air, are recommended in many different guidelines. In the ESGE guideline published in 2018, experts consider that outbreaks connected to insufficient drying on storage were mainly reported when instructions for drying were not followed, and that storage in non-controlled environment should prevent any secondary contamination. They also indicate that if non-vertical storage is chosen, special attention will be needed to ensure that no residual moisture will cause recontamination of the endoscope. So all experts agree on the fact that drying is really a key point, and that some things need to be done to eliminate water from endoscope channel. In another document published in UK, the HTM 0106 guideline, published in 2016, two options are provided for endoscope storage. The first one is to use a storage cabinet without drying function, but in that case, you have to dry manually your endoscope before storage. And we know that endoscope drying is something very, very complicated. The other option is to use a drying storage cabinet consistent with the EN standard 16442. And the main characteristic of this process is to passing air through all endoscope channels. So two options to avoid recontamination of the channel during storage. Today, a lot of these control environment storage cabinets are available on the market. But apart from this system, there is alternative technology such as plasma typhoon, vagascope, or shear store. Some of them are designing to dry and store endoscope, like the plasma typhoon. Some other are just made to store endoscope and store endoscope in a very controlled environment, like the shear source system. These processes using chemicals for drying and maintaining the quality of endoscope during storage are excluded from the scope of EN 16442. Nevertheless, manufacturer claims in terms of drying and control of the quality of endoscope must be verified. And the method proposed in the European standard for evaluation of the performance of drying storage cabinet can be used to verify such claim. As is stated in the French guideline published in 2016 and also in the HTM 0106, that such systems shall be submitted to the same validation process than drying storage cabinet complying with the EN standard. And the need to submit this process is well accepted by all experts. So in fact, as it is mentioned in the HTM 0106 document, all systems intended to provide a safe environment for the endoscope during storage or intended to dry the endoscope need to be type tested to verify and validate their effectiveness. But we also need to validate periodically the performance of such system as it is mentioned in the EN standard. And it's exactly what we have done with the plasma typhoon system. We have done some tests to verify that this system is able to dry endoscope and maintain the quality of the endoscope. The principle of the test is very simple. In fact, we contaminate two endoscopes with about 1,000 bacteria. Then one of the endoscopes is dry with medical grade air and stored horizontally. And the other one is dry and stored using the plasma typhoon system. As you can see with the test done with the PENTAX, for the endoscope, dry with medical grade air and stored outside the plasma typhoon system, we obtain a reduction of the contamination of about 1 log, no more than 1 log. And if we use on the other side the plasma typhoon system, what we observe that we can reduce the contamination by at least 2 log for one assay and around 2 log for the 2 other assays. And definitely this result confirm that the drying unit typhoon, of course, when it's used in accordance with the instruction for use of the manufacturer, allows a better elimination of the liquid present within the endoscope channel and therefore ensure a better control of the residual contamination of the endoscope. The same kind of test was done with so many different endoscopes. I just give you here an example of the coloscope, a Hygiene coloscope. And as you can see, the result obtained with the plasma typhoon system was really good. And by using this system, you can control the quality of the endoscope because you always have a reduction of the number of bacteria inside the channel for whatever contact time you consider. On the opposite, if you store an endoscope outside such system, without outside a control environment, what we observe is that we have a proliferation of the bacteria inside this channel. It is something really, really important. So, as a conclusion, what we can say is that the plasma typhoon and the plasma bag system have at least the same efficacy than conventional storage drying cabinets in terms of drying of internal channel of endoscope and maintaining the quality of the endoscope. Such a system presents also some advantages, like, for example, the drying time. You just need 1 to 5 minutes to dry an endoscope and therefore to avoid any biofilm formation. We also have a very long validated storage time because all the data presented demonstrate that you can store in a safely environment up to 31 days your endoscope. And the other important point is, of course, the fact that this system provides a safe and controlled environment when transport of endoscope is needed from one place to the other. Thank you very much.
Dr. Pineau, thank you for this overview. And drying and storage of reusable endoscopes obviously of its great importance. Why did you exactly choose to evaluate the plasma typhoon and the plasma bag system with regard to drying and storage?
Well, this is an excellent question. In fact, many innovating systems like the plasma typhoon, for many of these systems, it's quite complicated for the manufacturer to demonstrate their ability to do something, because most of the time they are outside any standard. In the case of plasma typhoon and plasma bag, it was quite simple, because the main objective of this system is to eliminate the risk of biofilm proliferation inside endoscope channel. So we decided to use the existing guideline and standard, and there is at least few of them, like the French guideline or the ESGE guideline, and of course the EN 16442, which provide us with few test methods to evaluate the ability first to dry and then to maintain the quality. So when they asked me what kind of method we can use to evaluate those processes, was quite easy just to adapt the method developed or present in the EN standard 16442.
So right, so you put the biofilm kind of in the center as a very important issue with regard to scope contamination, and the prevention of biofilm as kind of pivotal. How exactly kind of fits plasma typhoon and the plasma bag into that prevention of biofilm formation, and how do you then kind of see how it will improve the future of endoscope reprocessing?
In fact, the answer is in your question: drying, then drying, then drying. The key point definitively is to eliminate the water from the endoscope channel. And we know that, and we demonstrate with the test, that the plasma typhoon is very strong and very fast in drying efficiently the endoscope channel. So in fact, by making this drying stage much more efficient and easy to achieve, we will greatly reduce the risk of endoscope contamination and, of course, the risk of biofilm formation inside endoscope channels.
Thank you, Dr. Pineau, for these insightful answers. Ulrike, I want to have your point of view, because it's also pivotal for endoscopy nurses to be involved in this. What is, in your experience, the problems that can exist with drying endoscopes?
Drying requires, as Lionel really requires time and power. And time is always short in endoscopy. And from my experience, the drying, and that is also what the field studies showed, is often skipped. And then it causes problems with biofilm and the growth of bacteria. So we really need a standardized standardized drying procedure.
Right. So water is kind of an enemy if you want to do safe endoscopy after cleaning a scope.
Yeah.
How do you then foresee within an endoscopy unit, with the busy practice, how the plasma typhoon and plasma bag system would fit in? How would it work in such a environment?
I think the advantage is really that it is a standardized procedure, standardized drying time. So that is really the advantage. And you have two options to use it. Once just for the drying procedure, and then you have the other option for storage the endoscope. I think that nicely fits together. So that is really an advantage of the system. It is quick and easy with the drying, and then I have a safe environment for drying, for storage the endoscopes.
Okay, thank you very much for your answers, and thank you very much, Ulrike, and also Dr. Pineau, for giving a very insightful presentation. So in this symposium, we have learned about the advantages of using disposable consumables in endoscopes, like valves, brushes, and the removable distal caps. And in my view, these innovations have already contributed to lowering the chance of endoscope contamination and the possibility of patient to endoscope to patient infection. But future innovations may further improve the safety profile of reusable endoscopes, including, for example, removable working channels. We've also learned that there is still much to be improved with regard to sticking to manufacturer instructions and cleaning and allowing the cleaning personnel enough time to clean scopes properly. We've also learned that drying of endoscopes is of pivotal importance, since wet endoscopes and working channels are the ideal feeding ground for bacteria and biofilm formation. And plasma typhoon and plasma bag system are designed to deal with that issue and to swiftly dry scopes to prevent bacterial growth and biofilm formation, and to store scopes in a safe and secluded environment. Single-use endoscopes are already available and will make their way into the endoscopy suites of the gastroenterologist. However, there are many hurdles to be overcome, including economics, for this to take place. It's also expected for that reason that reusable endoscopes are there to stay for many years to come. And that highlights also the importance why companies, endoscopists, endoscopy nurses, and clinic personnel should keep paying attention and put all their efforts in improving the safety profile of reusable endoscopy. And with that said, I would now like to ask our viewers to participate, and we will start our question and answer session. Okay, and now we are live again, and I would like to welcome all the people that are just watching this session. And it's my great pleasure now to engage with the people that are watching, but also with our two presenters with regard to these presentations. Maybe my first question, Ulrike, just to kind of open up the discussion. We have now been talking so many years. We have publications, we have these kind of sessions talking about improving patient safety in endoscopy. You and Lionel have talked about product guidelines, improved instructions for use. Is there anything you can think about what we should do more with regard to trying to improve safety?
I think the first thing is that we need enough staff and also trained staff. So training is really a key issue, and we also need time for the reprocessing. And then, of course, clear instructions from the manufacturer, so short IFUs, that would be very nice.
Right, and it means also a message to the hospital administration people that they have to allow us to employ people to do this job properly and to clean these scopes.
Yeah, definitely.
A question that comes up in the chat is about the fact that, albeit we are talking about this for many, many years, it seems that we are not kind of reaching people properly with regard to the message, how important it is to do this, to clean the scopes. Is there any way we as professionals, or maybe the ESGE, should go about trying to kind of spread the message of the importance of scope cleaning?
I think the key issue is communication, that we have a good communication within the hospitals between the hygiene departments, the physicians and the nurses. And in case you have an outbreak, that is really an issue, that the communication is working. And you had this experience in your hospital. Yeah.
Absolutely.
For the societies, it's also communication. We have to write and communicate the guidelines. That is the point.
Right. I find it a little bit concerning that I still find people, colleagues, that kind of negating the problem. They think the problem does not exist. It only happens with the neighbors, who don't do the job properly. But in their own institution, everything is fine. Yeah. That's a little bit concerning. Yeah. Lionel, I am intrigued about this subject of biofilm formation in scopes. I've been reading about it, and there's nothing so difficult to understand as biofilms and how to deal with that. With regard to the plasma typhoon, and I get your message with regard to the importance of drying, but does that mean that you need to have a scope that has no biofilm, and then you prevent the biofilm from kind of getting there? Or can you also clean a biofilm with plasma typhoon? What's your view on that?
It's a good question. To answer the second one first, to eliminate biofilm from an endoscope channel, it's very complicated. Most of the time, you need a very strong cleaning agent, or even more, mechanical action is required most of the time to get rid of this biofilm inside the channel. But the most important, as you mentioned, is when you have a clean endoscope, and of course we have many of them, not all of them have biofilm inside, but when you have a well clean and disinfect endoscope without contamination inside, the fact that you are able to dry very in only few minutes in a well-designed procedure, as Ulrike mentioned, is definitely a good solution to eliminate biofilm formation. Biofilm grow only on endoscope where you maintain humidity inside and when you have a contamination, of course, at the beginning.
Right. But it does allude to the fact, kind of the same as we had a discussion with Ulrike with regard to the reprocessing people, that if you then employ the plasma typhoon system, you have to be very cautious. And you have to use it regularly with every step procedure, and you have to stick to the rules. Exactly. Otherwise, you still run the risk of biofilm formation.
Exactly, it's perfectly right. There is always a risk because we have water everywhere in the reprocessing room, and of course we have bacteria in the rinsing water. And if you maintain these two elements inside an endoscope channel, you increase the risk to have a biofilm formation inside your endoscope.
Right. You kind of nicely showed some of the preliminary work with regard to showing the efficacy of this system in kind of lowering bacterial load and exposure, which I think, you know, that is starting to be convincing evidence that we are doing something good with regard to trying to prevent bacterial contamination. But what now do we have to do to convince our colleagues, my colleagues, and decision makers with regard to installing these systems in hospitals and not just relying on the old way of dealing with scope reprocessing?
I think that the most important probably it's to making sure that people know exactly what's happened in their endoscope. And most of the time they are not aware about the real problem, because if we just speak about endoscope sampling, there is only few data published in the literature about the quality of the endoscope. And I think that one of the most important points is to have quality indicator like endoscope sampling, for example, just to highlight and to demonstrate that in some situation there is some contamination which are not, even some people don't know, or they don't feel that they have a problem. So
Right.
Quality indicator is definitely a good option to make sure that people may understand what's happened really in
I
their
think what
units.
you just said is a good point. If you don't recognize the problem, if you don't believe that you might run a risk, you will not notice the problem if you don't test for it. Huh?
Exactly.
Ulrike, you talked about the single-use consumables using in the reusable scopes, and of course the ultimate solution is the single-use scopes. There is this issue about weighing the costs of these type of devices versus the gain that we have. Is there anything you can allude to that? Can you say something about that?
If you want to calculate the cost, you have to put all the costs into the basket and say you have also for reprocessing not only the cost to buy the instrument, but also to reprocess it, and that costs a lot of things. And if you want to calculate that, you have to put all the costs of staffing, of training, machines, chemicals, all these things you have to take into account. And
it's incredibly difficult.
Yeah, it is. Yeah.
And I can tell you, if you had one outbreak, in my case with 30 patients, that costs us a lot of money to deal with the problem and to find solutions for that. There are some questions from the audience, and I think I hear these questions often, and I think they're very valid questions. That's about the environmental impact of these solutions, the consumables, the disposables, because it's waste that we throw in the bin, and what happens with it. Is there any concern about that? Do you know anything about the industry being concerned and thoughtful about this?
I think that we need their further research and logistics, but on the other hand, we have contaminated equipment, and I think to speak about recycling in a hospital is quite difficult. We can start with recycling material in our private life, like in the supermarket and these things. So I think speaking about recycling in a hospital, it's quite difficult, I think starting with the patient safety is more important than speak about the recycling of these things.
I think that's true, but I do believe that the industry is very well aware that for the success of implementation of these type of innovation, for them it's incredibly important to focus their attention also on this environmental impact with regard. And the other thing is, I often think that in this discussion, what people forget is that what we are now producing with regard to waste, using the reusable devices, with all the cleaning fluids that we use, thousands of liters per year, that's also enormous impact with regard to the environmental imprint.
Yeah, but not only the just the chemicals, but you also have these brushes, these cleaning equipment, then the packing material, and so on. There's a big number of waste you cause just with reprocessing.
Right, but I do think that these are very valid comments from the audience, and I think the industry also has to focus their attention to this type of issues. An important issue with regard to what I always hear from colleagues is that they try to save money with regard to the maintenance of scopes, and they try to avoid the official channels with regard to scope maintenance because they're mostly more expensive, and they go to these types of secondary industries that will do the maintenance of scopes. Do you recommend that? Is that a good cost-effective way of dealing with your scope maintenance?
It depends on the quality of these secondary manufacturers, and some use original materials, some not, but you really have to evaluate that. The gold standard should really be to use the original manufacturer because they have the experience, they have the validated processes, and also the original things,
Right. With regard to the FDA, they have recommended to use disposable devices and to turn our attention towards that. And also, we have now these single endoscopes being approved by FDA. How do you foresee that the introduction of these single-use endoscopes in Europe, will that fly in your opinion, or will we still have to focus on these reusable scopes and proper ways of cleaning those scopes?
I think there is already the trend towards single-use components. We started that with these single-use valves and also these single-use distal tips. And I think within a few years, we will definitely change to single-use endoscopes. It will take some time, but I think there is definitely the future for single-use endoscopes.
Okay. Lionel, with regard to prevention of biofilm formation, you have talked about the plasma typhoon, the plasma bag system. Is there any other way that people can behave or act with regard to prevention of biofilm formation? Because it's so central in the whole issue of scope contamination and patient infection.
Of course, there is so many things which can be done, and it's already mentioned in several guidelines. For example, the first point could be the selection of a good detergent, because we know that the quality of the detergent used to reprocess endoscope are really different from one to the other. And definitively, we have a good test method now to evaluate cleaning efficacy. We have a new standard, 15883.5, mentioning what should be done in terms of evaluation of cleaning. So if I have to select one answer, it's the cleaning detergent, or the detergent selection first, and then of course the second one definitely is drying and store endoscope in a well-controlled environment to avoid proliferation inside channel.
Right, and I think maybe as a last remark, although your high-level disinfection and plasma typhoon and plasma bag, I think, are valid solutions and important mechanisms, I think we should not forget that the key of scope cleaning still remains the manual pre-cleaning steps that have to be processed in a proper way. Otherwise, all these advanced systems can never do their work properly.
Exactly.
Okay, with regard to timing, it's now 7:45. We have come to the end of this symposium. I would like to thank Lionel Pineau and Ulrike Beilenhoff for a very interesting presentation and discussion. I want to thank the people that have taken the time at this late time step in the afternoon, in the evening, to be present. I thank you for your attention, and I hope to see you soon, maybe tomorrow or day after tomorrow, another session of ESGE Live 2021. Thank you very much.
Thank you.
Thank you.